Today we are going to discuss about the gene regulation, how the genes are regulated. As you know very well that we have millions of cells in our body, and each cell has got a complete set of genes, and all these genes may not express that particular point of time. Some of the genes will be expressed and some of the genes may not be expressed. And the genes which are essentially they will express to produce their products. That particular point of time, that product is essential for the body. That's why the genes are expressed. And some of the genes always they express. Suppose let us take example, there is a heartbeat. Heartbeat is essential for every second. That's why this heartbeat is under the control of particular set of genes. Those genes are always will be expressed. That type of genes, what we call it as constitutive genes. And the genes which are essential only that particular point of time, that only will be expressed, that product will be produced. So today here, how the genes are regulated, how the gene products are produced, that we are going to discuss in this class. Now let us see the first one. Virtually every cell in our body contains a complete set of genes. Just now I told you 46 chromosomes are present in every cell, and here 44 chromosomes are autosomes, and then one pair is a sex chromosome. All these set of chromosomes will be present in every cell in our body. That is one. Second one is, but they are not all turned on in every tissue, because these genes may not be turned on. Just like whenever light is required, we put on, then light, and then put off, light off. So just like that, genes also, when the gene product is essential, then the gene will be expressed. If the gene product is not essential, that particular point of time, that gene will be masked, it will be shut off. So like that, each cell in our body expresses only a small subset of genes at any time. Anytime that small subset of genes will be expressed. As I already told you, that type of genes, what we call it as constitutive genes. And then during development, different cells express different sets of genes in a precise, regular fashion. It is not haphazard. It is a regular, precise manner. The genes will be expressed and the gene products will be produced. Gene regulation occurs at the level of transcription or production of mRNA. You know very well that entire genetic information is embedded in the DNA. Deoxyribonucleic acid, you know, genetic material. In that, the information is embedded, and this embedded information is copied by mRNA. That's what you call messenger RNA. Actually, genes and chromosomes, you know very well, these are all present in the nucleus of the cell, and inside the nucleus, this mRNA will rewrite the language of DNA and then forming the transcript, and then it comes out of the nucleus into the cytoplasm. A given cell transcribes only a specific set of genes and not others. So this mRNA will rewrite the language of DNA. How much DNA information is required, that much information only will be picked up in the form of transcribed mRNA and will be coming out of the nucleus. So for example, insulin is made by pancreatic cells. Genetic information always goes from DNA to RNA to protein. That is what you call central dogma. DNA information is copied by mRNA. That is what you call transcription. And then mRNA, again this is translated by tRNA. That is what we call it as a translation. And after translating the nucleotide language of DNA into the polypeptide language of protein. So that is what you call it as central dogma. What you are seeing here, insulin is synthesized by means of this central dogma. And the gene regulation has been well studied in Escherichia coli. You know very well that bacteria is a very convenient material where we can study this genetic formations and the genetic expressions, and all these things can be studied because E. coli bacteria contains very limited amount of DNA. It's very easy to manipulate, or you can say study limited genes rather than we have 45,000 genes in our body. It's very difficult in eukaryotic cells. That's why for genetic studies, generally we take prokaryotic organisms like Escherichia coli bacteria. When a bacterial cell encounters a potential food source, it will manufacture the enzymes necessary to metabolize that food. Now the food is available for the bacteria in the medium. So that food is how it is utilized, how it is metabolized by the cell. So it requires varieties of enzymes. When the food is available, it will try to synthesize the enzymes which are essential for digestion of that particular material or food material. That type of enzymes will be secreted and that type of enzymes can be produced. Unless that protein is not available, some food is not available, these enzymes are not essential. That's why enzymes will not be produced unless the food material is available. That means here a lot of conservation of energy, conservation of activities can be seen in prokaryotic organisms like bacteria. Now here you are seeing ultimately today the important discussion in this class is Jacob and Monod, famous geneticists. They have introduced this model, Jacob and Monod model. They explained how a gene expression can be regulated. How the gene expression can be regulated. What are the factors? How the gene product is produced. How it is useful to the organism. That is discovered by, it's a famous discovery, wonderful discovery, and after their discovery only we came to know that one gene, one enzyme, one gene, one polypeptide, one gene, one character, all these postulates were formulated by the post-Jacob and Monod scientists. Now here Francois Jacob and Jacques Monod worked out how the lag operon functioned and they formulated the operon hypothesis. Now this lag operon concept, famously it is called as lag operon concept because Jacob and Monod, they have discovered this famous model by taking this bacteria. Let us see this one, Jacob and Monod method of regulation of a gene. Now you see here, Jacob and Monod proposed the lag operon concept to explain the regulation of the synthesis of an enzyme beta-galactosidase by E. coli only when lactose is available as a substrate and glucose is not available. This was the first study of metabolic regulation based on genetic analysis of the lag system. They called this the operon model or lag operon concept. The basic concept in this lag operon is lactose is a disaccharide. This lactose disaccharide is used by the bacteria. E. coli bacteria is using this lactose, that is disaccharide, and after taking this lactose into the body, that lactose can be metabolized for production of energy. When the lactose is metabolized, two simple products are produced, that is glucose and galactose. Glucose and galactose. And these glucose and galactose are very simple units that can be absorbed very easily in the bacteria. At the same time, the essential amount of energy is being released. That is the important thing here we have to remember. Now the question is whether the lactose is available in the medium or not. If the lactose is available in the medium, then there is a need of production of this beta-galactosidase enzyme, which is very much essential for metabolize this, or you can say hydrolysis of lactose to form glucose and galactose, that is monosaccharides. Now here the genes which are present in a cell express to prepare a particular function or set of functions. Beta-galactosidase enzyme is synthesized by E. coli bacterium to catalyze the hydrolysis of disaccharide lactose to form galactose and glucose. These monosaccharides are used by the bacteria as a source of energy. As I already told you, these two monosaccharides are utilized for metabolic activities. At the same time, sufficient amount of energy is being produced. That is main purpose of this beta-galactosidase enzyme. So beta-galactosidase enzyme is produced in presence of lactose only. If the lactose is not there, there is no need of this production of this particular beta-galactosidase enzyme. If the lactose is not available for bacteria, there is no longer requirement of enzyme beta-galactosidase. By this example, we can understand that metabolic, physiological, environmental conditions that regulate the expression of genes. Now, the expression of genes is influenced by environmental conditions, physiological conditions, metabolic conditions. Basing on these conditions, this beta-galactosidase enzyme is produced. Now, coming to this lac operon, gene expression leads to the formation of a polypeptide. This can be regulated at different levels. In eukaryotes, the regulation could be in the following levels. It's very, very important. You know, we are talking about this enzyme, beta-galactosidase enzyme. This enzyme, you know very well, all enzymes are proteins. All proteins are not enzymes. You know very well. Now, beta-galactosidase enzyme is a protein. To synthesize this protein, whether the lactose is available or not, that is a conditional thing here. Now here, in this synthesis of this particular gene, which is essential for synthesis of the beta-galactosidase enzyme, there are 4 important levels. One is transcriptional level, second one is processing level, third one is transport of mRNA from nucleus to the cytoplasm, translational level in cytoplasm. Now you see first one, transcriptional level. Transcription, as I already told you, that DNA segment of DNA which contains all the codes. These codes will code that particular protein, that is enzyme. So to prepare mRNA, the transcriptional level, how much amount of segment should be transcribed, what length of DNA should be transcribed, that is here. This is the first level of control. And the second level of control, processing level, regulation of splicing. You know very well exons and introns. In a DNA, exons and introns are there. From the DNA, when the mRNA is synthesized, complete polynucleotide is converted to polysaccharides. Polypeptides. This polypeptide, some of the polypeptide may code, some of the polypeptide may not code. So that is what we call splicing, exons and introns. In this level also, processing level also, it will be regulated. Transport of mRNA from nucleus to cytoplasm. So from the nucleus, because transcription has taken place in the nucleus on DNA segment, then after meaningful mRNA, this mRNA should be transferred to the cytoplasm where the protein synthesis must be taken place. That is the transport of mRNA from nucleus to the cytoplasm. Next one is translational level in cytoplasm. So after transcription, next step is a translation where mRNA should be translated. mRNA is having all the codons which are taken from the DNA. These coded language should be converted to amino acid language. That is a protein. Proteins are made up of amino acids. Now, polynucleotide language should be converted to amino acid language in a protein. So, when the protein is synthesized, how it is controlled? That is the fourth step, translational level of control. That is in cytoplasm, it takes place. Now, RNA polymerase is also an enzyme. This RNA polymerase is helping for the transcription of the structural genes. Structural genes must be translated in the form of mRNA, this mRNA. So, RNA polymerase is essential for synthesis of mRNA from the structural genes. So, 3 structural genes you are seeing here, LAC-Z, LAC-Y and LAC-A. 3 important structural genes must be transcribed in the form of mRNA. This mRNA further translated to form beta-galactosidase, permease and transestirase. All genes can't express at the same time. Some genes are needed for the function of all cells all the time. These genes are called constitutive genes and are expressed by all cells. Previously I have explained this constitutive genes. Always they express because those genes products are very much essential for metabolic activities of organism. That's why always they express. Likewise, other genes are only needed by certain cells or specific times. The expression of these inducible genes is tightly controlled in most cells. For example, beta cells in the pancreas make the protein insulin by expressing the insulin gene. So certain time, certain number of genes must be expressed. So that is what we call the other one. And the next one is operons are groups of genes expressed by prokaryotes. The genes grouped in an operon are all needed to complete a given task. Each operon is controlled by a single control sequence in DNA. Because the genes are grouped together, they can be transcribed together, then translated together. That is the core concept of operon transfer. Core concept of operon. Core concept of Jacob and Monod. Here, what I mean to say, group, that group of structural genes, what we call it as operon. Operon including regulatory gene, operator gene, structural genes, and all these together what we call it as operon. These operon completely they work together. In a group they work. That's why it is called as operon. Let us see next slide. We are going to see that structural aspects needed to complete a given task. Each operon is controlled by a single control sequence in the DNA. Controlling sequence will be there. That's what we call it as regulatory gene. Because the genes are grouped together, they can be transcribed together, translated together. Now you can see genes in the lactoferrin allow Escherichia coli bacteria to metabolize lactose. Lactose is a sugar that E. coli is unlikely to encounter, so it could be wasteful to produce the proteins needed to metabolize it unless necessary. If the lactose is there, then enzymes are produced. If the lactose is not there, production of enzyme is a wasteful process. That's why this wasteful process will not be initiated by prokaryotes. They conserve the energy and the materials. That's the second part. Metabolizing lactose for energy only makes sense when two criteria are met. Other more readily metabolized sugar, glucose is unavailable, lactose is available. Now here promoter is there, operator is there. These are the specific DNA sequences to which RNA polymerase binds so that transcription can occur. The lag operon promoter also has a binding site for another protein called CAP. Operator, the binding site of the repressor protein, the operator is located downstream, 3' direction from the promoter so that repressor bound RNA polymerase can transcribe. Actual diagram just you see here. Now here repressor is there, cap is there, promoter is there, operator is there, lag Z, lag Y, lag A. So lag Z, lag Y, lag A, these three are structural genes. These three are structural genes. And then this one is operator gene. And this one is a promoter gene. And cap is there and repressor is there. Now let us see when glucose is present but not lactose. Now let us see how it is happening. I am constitutive. Repressor mRNA. Repressor mRNA produces repressor. And this repressor will go and bind to the operator. It's a blocking. Operator gene is blocked now. Now RNA polymerase, let us see. Come on, let me through. No way, because its operator gene is blocked. Now this synthesis of this lag Z, lag Y. Now, no way goes. That means here lag Z, lag Y, lag A will not be transcript. No transcription. When there is no transcription, no